All right, good morning, students. And today we are going to start with another class of motor proteins called dyneins. And honestly, this is going to be the first time I'm going to teach something which I haven't really worked on. So, dynein motors are something that I've never worked on. Also, dyneins are something that are highly complex compared to myosins or kinesins. These motor proteins are highly complex and very large. They have associated chains which themselves can be multi-subunit complexes such as dynactins. So we'll try to parse through dyneins in a couple of classes, and today is the last class before your minor. There will not be a class on Friday. So what I'm going to do today is just give you an overview of Dynein. Before I start the class, can you see the slides that I have shared? Is it visible to you all?
Yes, ma'am.
Yes, okay. Let's. Okay, so the existence of dyneins has been known for almost, I would say, 70 years now. But it's only very recently that people are beginning to understand how dyneins function, as in how the structural elements come together to make this motor work. And yet there is a lot that is not known about dyneins. So this is kind of the first paper, you can see 1963, where dyneins have been described. And what you can see here on the right is an image of a single-celled organism. And you see that the body of this organism is covered with um, a cilia or flagella, whatever you call it, those small appendages that is going to help this organism move in its environment. And what was done in this paper was that they took these appendages and extracted the membrane from the surface of these appendages using a very mild detergent. So the mild detergent that they used is called Dynein, and very often to understand subcellular cytoskeleton structures, we use uh, this mild detergent. What it essentially does is it kind of creates transient pores on the membrane. And if you treat it long enough, of course, you would be able to create larger pores and, in fact, remove the membrane. But because this is a mild detergent, it is not going to interfere with the way the proteins are arranged. So, using this detergent, they were able to observe the structure inside these cilia. And what they found was that if they took now this detergent extracted cilia and they made a section out of it and observed it under the electron microscope, this is what they could see. Now, this structure is not unknown to those who have done some amount of biology in their masters or in their graduation days. So what you see is these ring kind of structures all around, the 9 rings, doublet rings, and in the center you may or may not have in the center 2 additional rings. And these rings are made up of microtubules, right? They are tubulin structures, microtubule structures. So let's go back to this. So you can see these doublet rings, 9 of these, and in the center you may or may not, in some organisms you do, some you may not, have these central doublet tubulin rings. And the way it is thought, I mean, it's still not absolutely clear, but it is thought that the way these would function and the way these would help the appendages move is by sliding these microtubules past one another. So the sliding motion is supposed to be moving the appendage. Now that's quite a difficult thing to kind of envision, that how a sliding of microtubules past one another is actually going to create a whip-like motion. As I said, not everything is known, but it's still believed that this is how it is working. And how does these microtubules move past one another? The way they move past one another is with the help of motor proteins. And these motor proteins are not kinesins, but dyneins. And in this paper, what they did was they identified these. So if you can see in this image, you would see that there is this semicircular kind of a structure here at the end of each of these doublet rings. So this semicircular kind of structure is made up of dyneins, the outer dynein and the inner dynein arms. And these dyneins are supposed to make contact with the next doublet microtubule and help in the sliding motion. So this paper was actually the first one to show that there is the existence of dynein proteins. They were able to sort of purify the dynein protein and show that this is a very large protein. Protein. It's about 500 kilodaltons or more. So that's huge. It's a huge protein. And they were able to show that there is also dependency on ATP, just like kinesins. These motor proteins also depend on ATP for their function. Let's move a little ahead. So this, what they saw here is what, I mean, if we refine the structure, this is what it comes to. You do not have to sort of remember all the elements here. We are not quite interested in looking at each of these elements, but just to give you an idea that this is, again, what we are talking about, these appendages, they are made up of numerous proteins, numerous cytoskeletal proteins, prominent among them being the tubulin, the microfilaments here, the microtubules, the tubulin structures here and here, and the dynein. So these are the outer and the inner dynein arms that are known to make contacts with the next microtubule doublet. Okay, so then the question is, where is dynein present? I mean, we have seen the first paper where dynein was extracted or identified in cilia. So where is dynein present? So dynein is present, of course, in cilia, in flagella, and in all of their motility. You would require dyneins. Not flagella of bacteria, don't get confused, eukaryotic flagella. So for example, sperm flagellar movement, that is dependent on dynein. But dynein can exist in cells other than these appendages. So you would have something called a cytoplasmic dynein. Now, if you remember, in the previous classes, we have always said that conventional kinesins move on microtubules and they move towards, from the center of the cell towards the periphery of the cell. So they move towards a plus end. So that's conventional kinesins, right? We know that kinesins can also move towards the negative end. Kinesins can be dismantling microtubules. So all of that we know, but conventional kinesins will move towards the plus end. Dyneins, on the other hand, they move in the opposite direction. So when we said this, that the dyneins move in opposite direction, we were mostly talking about these cytoplasmic dyneins. So there are some dyneins which are present in these flagella or cilia, these appendages, and they are fixed in their space, and they help in moving past these microtubules, one past the other. But there are also cytoplasmic dyneins which help in carrying cargo from the periphery of the cell towards the center. So you have kinesins that move from the center towards the periphery. You have things that have to be moved from the periphery towards the center. That's the dynein function. So dyneins, in this case, are doing a minus-ended walk, right? Even in the flagella, the walk is minus-ended at the end, but then they are fixed in space. Here, they are actually moving with the cargo towards the minus end. So dyneins essentially do a lot of things. They move, they can be involved in movement of cargo. Cargo could be anything. It could be just like kinesins, it could be movement of organelles to movement of ribonucleoproteins, movement of vesicles. It could be a number of things. It could also, they are also required during mitosis, where they function at the kinetochore complex, and they help in movement of the microtubules and they help in dismantling of the nuclear membrane. So that there are hundreds of functions that they do. And okay, let's go back to this slide. So because they are involved in so many processes, and the one very significant process being the movement, the flagellar movement or the ciliary movement, they are associated, or defects with Dynein function is associated with something called ciliopathies. We'll come to this later after the minors. We'll come to all of these myopathies, ciliopathies. But it gives you an idea. So if you have a motor protein that is required for the motion of these appendages, and if there is a problem with the motor protein, then definitely there would be a problem in moving these appendages. So problem in movement in appendages might mean problems in movement of the sperm, or it might mean problems in the air passage, because our airway epithelium is lined by these appendages that move, right, that beat. So if there is a problem in Dynein, then that beating motion would be affected. Okay. Now, then we have seen where dyneins are present. We have had a brief idea that what are the functions it could do, but how does it look, right? That's the next question. How does a dynein look? So unlike myosins and kinesins, they more or less, I wouldn't say they looked similar, but at least they had some commonalities between them. But dynein is a completely different class of protein. And what you see here is something called triple A ATPases. Now, triple A ATPases, what are these? These are proteins that are found in almost everything you know. You take bacteria, you take viruses, you take human cells, you will see these classes of proteins. And what these classes of proteins does, they are involved in a lot of processes. For example, they are involved in unfolding of proteins. They can be involved in unfolding of RNA or DNA double helices. For example, some of the helicases involved in, say, bacteria, or even in viral replication, you would see that they belong to this AAA ATPases. And as the name suggests, it's an ATPase. That means it requires ATP for its function, right? And we saw that the first paper which dealt with dynein, which had identified dynein, they have also said that it requires ATP for its function. So here is a class of protein called AAA ATPase. It's involved in diverse functions found in all kinds of organisms, from bacteria to virus to humans. And if you look at this is one unit of a AAA ATPase. And if you look at this very carefully, you will see that it has this small subdomain, or a small domain, and a large domain. And the large domain is where the ATP binds. Okay, so this is AAA ATPase. I've not come to dynein yet. This is just AAA ATPase. It has a small domain and a large domain, and the large domain is binding to ATP. But this one unit doesn't function by itself. It oligomerizes into a ring-like structure with at least 6 subunits, which are arranged in such a fashion that the binding of ATP here and the ATPase function requires the help of this subunit from the next, requires the help of residues from the next subunit, so from here. So binding of ATP and ATP hydrolysis here requires the help of the subsequent subunits. And so this is essentially one functional unit of AAA ATPase, where each subunit is able to hydrolyze ATP and is able to function in, say, unfolding of a protein or unraveling of DNA double helix. So all kinds of functions it does. Now, how does this correlate to dynein? What dynein has done is it's a very smart protein. It has taken this concept of these 6 subunits forming this ATPase and combined it into one single polypeptide. So instead of actually looking at 6 different subunits, one dynein protein has these individual 6 subdomains. Each of them comes together to form a ring-like structure, as you can see here. This is essentially equivalent to this. So if this is equivalent to it, which means that each of these subdomains in dynein would have an associated ATPase function, because it is, after all, it takes the whole idea of this AAA ATPase, converts it into a dynein structure, right? A dynein ring structure. So in reality, we do see that each of these subdomains of dynein has an ATP binding activity, but not all subdomains are equally responsible for ATP hydrolysis. So this is one difference between AAA ATPases and dynein. In the end, you do see that there are these distinct subdomains. 6 of them would come together to form that ring-like structure. Each of the subdomain has the smaller domain and a larger domain. And as I had explained, the whole idea behind having these two, why I'm trying to explain these subdomains is because you have to understand that the ATP binding, ATP hydrolysis activity of one region is dependent on the smaller subdomain of the next subunit. So if you translate this to dynein, then the same thing happens there. If you look at these different subdomains, then this subdomain 1, this AAA1, is the point where the ATP hydrolysis actually takes place. If you mutate residues that are binding to ATP in this subdomain, or mutate residues here that are responsible for hydrolysis of ATP, you would see that this dynein is not able to function. So, okay, so this is about the AAA ATPase kind of domain. So far, is it clear?
Yes, ma'am.
Okay, thank you. So, before we go
Yes,
into
ma'am.
any, yes, yes. Before we go into any further structural details, let us look at some things which are mundane, and I'm just going to read it out because it's good to know this information. Not that you have to memorize it, but it's good to know. So kinesins, we found that there were like 45 different genes for kinesins, right? Because kinesins have to carry diverse proteins. So, but dyneins also, they are doing a lot of different functions, right? They are present in the appendages, they are present inside the cytoplasm, they're carrying different cargo. So just like kinesins, dyneins also have to be very diverse. But the way dyneins achieve this diversity is a little different. So let's look at this. There are about 9 major classes of dynein heavy chain. Why dynein heavy chain? So, okay, so this one single structure that you see here, this alpha beta is triplet, but this one single structure you see here, and there is also an associated stop up to this, is actually one single very large polypeptide that folds into this structure. So we'll call this the heavy chain. Just like in meiosis we had a heavy chain, we call this the dynein heavy chain. So there are 9 major classes of dynein heavy chain. There is one class of this which makes up the cytoplasmic dynein. So all the dyneins that are involved in transporting vesicles, etc., inside the cell, that is done by one class of heavy chain dynein, and this is the name of the gene that codes for this cytoplasmic dynein. There is another class of cytoplasmic dynein which typically is not quite cytoplasmic. The reason it's called cytoplasmic dynein is because they help in movement of cargo, but inside appendages, inside flagella or cilia. That does not mean that the appendages that were attached to the tubulin dimers, not those. Those are integrated into the structure of the cilia or flagella. But there can be other appendages that move along. There could be other dyneins that move along up the appendage and down this appendage. Those are another class of cytoplasmic dyneins. So you have these two cytoplasmic dyneins, and the remaining 7 other classes of dynein heavy chains all go into building these axonemes or these cilia and flagellar structures. So they help in beating of the cilia and flagella. Now, axonemal dyneins can have multiple copies in the genome. Also, these dynein proteins could be, the dynein mRNA can be alternatively spliced. There could be phosphorylation and other post-translational modifications that give rise to certain amount of diversity between dyneins. So even though you have one single cytoplasmic dynein, by alternative splicing or post-translational modifications, you could give a little variety to it. But the major variety of cytoplasmic dyneins comes from the accessory proteins that associates with it. So remember, we have so far talked about the dynein heavy chain, right? There's one single very large polypeptide chain that folds into a structure more like this. This is like the cartoonic representation. But along with this one single folded polypeptide chain, you will have some associated proteins. For example, here we have listed some of these associated proteins called light intermediate chain, light chains. There are some associated proteins that attach to the dyneins and that modulate its function and modulate also its structure. For example, if you remove this light chain, you suppose you isolate dyneins, and you are looking at an in vitro motility assay of dyneins on microtubules. You would see that if you remove these associated light chains or light intermediate chains, instead of the dyneins actually moving on microtubules, you would see dyneins clustering together and not forming these distinct dynein motors. So these associated light chains, light intermediate chains, are required for the for proper formation of the dynein structure, its proper functioning, and also association with the cargo complex. Apart from these, apart from all of these cargo or adapted proteins that we have talked about, there are other of proteins it can associate with. For example, here there are names of the 3 associated proteins, of which I think dynactin complex is at least one which you must have heard about, because dynactin itself is a multi-subunit complex. It's not made up of one protein but multiple proteins, and together with all of these, the light chains, intermediate chains, and all these associated dynactins, etc., the functioning of dynein and the cargoes it carries are selected. So again, let me emphasize this. Dyneins is still something that we do not quite completely understand because it's hugely complex. It's hugely complex. And this lecture that you're listening to now, I'm sure if I carry on with this class for another 5 years later, this same lecture is going to change tremendously, because it is still a topic in evolution. Not much is known about it. Very little is known. Whatever we know is still very little. Okay. So let us come back to the structure of dynein, because that is where we are going to focus today. So we have time and again emphasized that the main structure or main heavy chain of dynein is one single polypeptide, but it has these 6 individual domains of AAA plus ATPase module. So you have these AAA plus ATPase module, right? 6 of them. 6 of them in such a manner that they can fold in the way of a ring, exactly like the AAA ATPase do in nature. They fold themselves in this ring-like structure. Its similar thing is achieved, but by distinct domains within the heavy chain. So you have these domains. Apart from that, on the N terminus, you have something called the tail region, followed by something called the linker region. And if you way this whole structure comes together, then this is more or less a cartoonic representation of how a Dynein heavy chain would look. And this is the way these heavy chains are, the domain structures are arranged on the polypeptide. So if you stretch the Dynein heavy chain from the N terminus to the C terminus, you find that at the N terminus you have something called the tail. We usually associate the idea of tail with the C terminus, but here the N terminus has the tail. And what the tail does is, it is the point that connects with the cargo. And in fact, very little is known about this. So most of the idea, the crystal structure, etc., that we have deal with this part, and this part is still a little obscure. But nevertheless, the N terminus is what is the tail. We will call it the tail, and the N terminus is what is going to be binding to the cargo. Followed by this, you have something called the linker region. And after the linker region, you have these 6 modules of AAA ATPase domain, right? And the 6 modules will come together to form a ring-like structure, as you can see here. Now, what the linker region does is very interesting. Its position is determined by the state of ATP binding in in this subunit mostly, AAA A1. Because AAA A1, as I said, is the main subunit that has an ATPase function. ATPase function is also associated with AA3, but if you mutate the ATPase function of AA1, the dynein is dead, whereas that's not the case with AA3. So it's from these mutagenesis studies that it has been identified that AA1 is the main region where ATP hydrolysis takes place. And the way the linker is positioned on this circular structure of the AAA subdomains is determined by whether this region is bound by ATP or bound by ADP and PI, or the PI is released or doesn't have a nucleotide, right? And in this sense, there is some amount of similarity with the way the power stroke was being performed in myosins. So if we go back to understanding myosins, and I know you have already explained in the last class how myosin, last or the previous class, how myosin ATPase cycle actually worked. Can you once more, one of you, please, can you once more repeat how the myosins function in terms of this ATP hydrolysis and creation of the power stroke?
When myosin had first at this time ADP phosphate. When phosphate is released, there is a power stroke and conformational change pulls myosin head away from actin. At this stage, ADP is also released and then ATP binds. And when ATP is hydrolyzed, conformational change again happens and head moves closer to the other end and it reattaches.
Okay, so the question here is that does myosin bind to actin when it is bound by ATP?
When it is bound by ATP, no, ma'am.
Okay, thank you, sir. Thank you for this
No,
explanation. So
ma'am.
we have
No.
yes, Farman. Yes, Farman.
Uh, Nothing, ma'am. Actually, there is some network problem.
Okay, okay. So So we take this whole idea now and we look at dynein and how it functions. Um, okay. All right. Before we go any further, let me see if I've missed some points. Maybe I have. I have already spoken about the diversity and how much we know about it. It's actually not too much that we know about it. I've said that, okay, it's a very large polypeptide, about 500 kilodaltons. Anything else? Um, Recruitment of adapters. Oh, okay. This is one thing that I have not mentioned. So as we have seen that this tail domain, which is at the end terminus, um, is required for binding to the cargo, right? And it is the point where many of these light intermediate chains actually bind. So this region, because it determines what kind of cargo is going to be carried by Dynein, all the accessory proteins that interact with Dynein and helps this Dynein to function in terms of carrying the cargo is actually regulated at this region, which is called the tail region. Now, if you remove the tail region, would the Dynein still function and work by binding to the microtubule? Ideally, yes. And in fact, it is the case. The minimum unit that is required to bind to microtubule, right, to bind to microtubule exists here. And you can literally leave out the tail. So just like kinesins, what were we doing with kinesins? We were taking the if you imagine the classical kinesin, you have a double helical structure with the microtubule binding end at one or cargo carrying end at one end, and you have the motor heads on the other, right? So the motor heads are actually walking on the microtubules, and you have the cargo carrying end at the other end. And if you remove this coil domain and the cargo carrying head, the rest of it still functions in binding to the microtubule. A similar thing, you translate that to Dynein. You cut off this, the rest of it would still bind to a microtubule. So this region is what is giving it selectivity towards the cargo. Not much is known about this region. It's still a little, not a little, it's a lot of obscure. We do know that it binds to all of these associated adapter binding proteins or intermediate chains or light intermediate chains, and it helps in selecting out the cargo. So that's all that we know about it. Okay. Now, if you look very closely at the structure once more, we come back to the structure once more. You see that you have the N-terminal tail, as we said. This is something that selects out the cargo. You have then a linker region, and we said that somehow the positioning of the linker is actually required in the movement of the Dynein, and we'll come to that in a bit. And then we see these associated 6 subdomains. And we said that the AAA1 is the main ATPase. There is some amount of ATPase activity associated with AAA3, but if you mutate AAA3, the Dynein functioning, the Dynein speed is still there, maybe it is just slow. But if you mutate the ATP binding site of AAA1, then you see that the Dynein becomes dead, it does not move. So this is the primary ATPase site. And between this AAA4 and AAA5, you find something called the stalk and microtubule binding domain. So essentially here is a sequence of amino acid that forms a coiled coil. Now you see the coiled coil has come back again if you remember kinesins, you had the microtubule binding end and the cargo carrying end, and both of them were connected by coiled coil, and the coiled coil was also helping in dimerization. Here the coiled coil is not helping in dimerization, although these cytoplasmic dyneins can form dimers. Instead, the coiled coil structure here kind of translates the changes associated with ATP hydrolysis to the movement of the microtubule binding end. So all of this is one single polypeptide. Again, let me emphasize that this entire thing is one single polypeptide. It's only a matter of how each of these subdomains folds into specific conformations. So you have between AA4, AA5, you have a region that forms this coiled coil and the microtubule binding end. So all of this is present within this heavy chain. And between AA5 and 6, you have something called the buttress, or something that extends out and makes a connection with this stalk. And you can see that by making a connection with the stalk, it kind of acts as a point where it can sort of regulate the binding to the microtubule. So it's kind of something that, suppose you have a region that moves, right, and you create a link that would stop it from moving any further. So it's kind of it helps to keep this stalk in kind of a position. Okay, now the whole field of Dynein has evolved because of our understanding of its structure, and the structural understanding comes from either extracrystallography data, or it comes from cryo-electron microscopy data. So both of them have gone hand in hand to give us an idea of how the dyneins function. So again, here is an cryo-electron microscopy image of a dynein. You can see that this is the AAA ATPase ring-like structure of the dynein. You have a region that extends out, and this is the stalk. This is the stalk that has this kind of a globular end that binds and grasps microtubules. Oh, sorry, this is the stalk. I get always confused. Right? So this is the stalk. And then you have a stem-like structure that would go and tail-like structure that would go and bind to the cargo. Okay. All right. So now we come to how we think that the dynein is able to function as a motor protein. And as I said, a lot of this understanding comes from X-ray crystallography structure or cryo-electron microscopy structure. So using or obtaining crystals of dynein, uh, of course without, sometimes even without the tail, but with different agents. For example, you could have a non-hydrolyzable ATP bound to dynein, and you could have a crystal structure of that. You could have an ADP bound dynein, and you could have a crystal structure of that. So looking at all of these crystal structures gives us an idea of how the dynein might function. So this in brief gives you a representation of how we think the dynein functions and works on microtubules. So if we think of a classical cytoplasmic dynein, we are talking of a double a dimer. So you have to now imagine two such dynein rings attached to the attached to the microtubule, two such structures. And if you now look at one such structure, this is the coil domain with the end that binds to the microtubule. And what you see associated here is the linker. And along with the linker, we know that there is the N-terminal tail that binds to the cargo, right? And when it is bound to ATP, when this the AA1 is bound to ATP, that's the point where the dynein doesn't make a contact with microtubule, and the situation is very similar to myosins. So when bound to ATP, it is not making a contact with the microtubule. But when the ATP is hydrolyzed to ADP and PI, that is when it searches out for the next binding site on the microtubule. Now, there's another interesting thing here. When you try to imagine there to be two dyneins, so suppose there are two dyneins, and both of them are attached to the cargo. There's dimeric dynein, both of them attached to the cargo, and they are walking on the microtubules, right? So it can be that one of them takes 2 steps forward before the other one actually moves. So dynein, unlike kinesin, does not have a very regular movement of one hand over the other over the other. Rather, dynein can have regular movement, can have a movement of one leg moving, say 2 steps, 3 steps before the other leg moves forward. Also, dynein can have one leg moving forward and then moving backward, and the other leg can move forward. So dynein motion, what I'm trying to emphasize here is that unlike kinesin, where you know that one leg moves forward, the other leg moves forward, it's very periodic. Dynein motion is not so periodic. And in dynein, the two dynein heavy chains can have a sort of independent movement of each other. And when they move on the microtubule, the two legs are actually attaching to two different protofilaments of the microtubule. So they're moving like this. They're very sloppy movement. It could be that one leg moves backward, the other leg moves 3 steps forward. So it's not as systematic as kinesins. So here you have a state where it is bound to ATP and it is not bound to microtubule. The ATP is hydrolyzed and it searches out for a binding site on the microtubule. It can have a weak binding site, a weak binding to it. The release of PI then induces a power stroke. Remember myosin, and you can see the analogy here. And that creates a change in the, what should I say? So the other thing that is important here is the position of the linker, and I'll come to that in a bit. So this creates a power stroke, and you can see that the myosin head would now go and move to a different second binding site on the microtubule. But this second binding site could also be at the back, because dynein, unlike kinesin, so for kinesin 2, for single kinesin heads, we have seen that sometimes the kinesin heads can move backward. But usually if you have dimeric kinesins, then this backward motion is inhibited and it kind of keeps moving forward. For dynein, the motion can also be backward even when it is in the dimeric state. It can take a step backward because there is a binding site for it at the back. So it could move backward, and then the other leg could move 2 steps forward or maybe 1 step forward before this other leg catches up. So dynein motion in that sense is pretty it's not symmetric in the way the kinesin movement was symmetric. Let me come to a better way to explain this. So you see, the whole idea of how the dyneins work and move, as I said, cryo-electron microscopy images have given us a lot of idea about how this dynein functions, and how they have worked around the problem of trying to understand each step of dynein function is to utilize dynein with different analogues of ATP bound to it. So here is a cryo-electron image of dynein, and you can clearly make out the tail and the stalk that binds to the microtubule, right? This is the stalk that is binding to the microtubule. You have the ring-like structure. This is the tail. So exactly what you're seeing here, you're seeing a cartoonic representation of this here. So you could have the dynein bound to a non-hydrolyzable ATP analog, something like this. Or you could have it not bound to ATP at all. So that is something like this. And what you see is in both these conditions, when it is not bound to ATP versus when it is bound to ATP, what changes is the way this stalk and this linker region here is positioned. So the positioning of the linker that actually falls on this ATPase ring. And what was the linker? The linker was this. So you have the tail right? You have the linker, and somewhere here you had the stalk. So positioning of this linker region and the stalk changes between the way the ATP analogs are. So if it is not bound by ATP, if it is bound by ATP, if the ATP is hydrolyzed to ADP plus PI, and if the PI is released. So at each of the state, you see that the linker position is different. And what this creates is, it creates a distance between this head, sorry, not the head, but the tail, the N-terminal tail, the distance between the tail and the ring. Effectively, what it does is it pulls the cargo along, because remember, the cargo is attached to the tail. So if you can change, so if this is the ring and this is the tail, and you change the distance between the tail and the ring, you're effectively pulling along the cargo. So each of this motion, apart from the fact that you have this stalk that makes connections with the microtubules and it is itself moving forward, you also have a motion of this region that is bound to the cargo, and depending on the ATP hydrolysis state and the positioning of the linker, this cargo is also being pulled along. So every time there is the cargo that undergoes a motion because there is change in the positioning of the linker. So the entire thing is quite complex even for me, but this is how I try to envision it. You have a change in the position of the stalk. And this positioning of the stalk is also determined by the coiled coil domain. Because it's a coiled coil domain, you can have changes in the relative orientation of the two coils, and that translates to how the microtubule binding end positions itself on the microtubules. And the binding on the microtubules, as I said, is not symmetric. So if you have these two Dynein arms that are walking on the microtubule, the two arms are walking on two different protofilaments. So there are 13 such protofilaments in a typical microtubule. You imagine two parallel protofilaments, and the Dynein arms are walking on two parallel protofilaments. You could have one arm seeking out one protofilament and moving, say, one step forward or two steps forward, but it could also move backward. And one of this arm could move two steps forward or three steps forward even before the other arm catches up. And the other arm could catch up and move past this. So it's very asymmetric, the movement of the two arms. And it is all coupled to ATP hydrolysis. So when it is bound by ATP, that's the time when the Dynein arm is not bound to the microtubule. Hydrolysis of ATP to ADP plus PI creates a weak binding state on microtubule, and release of PI creates a power stroke that propels the Dynein arm kind of creates the power stroke for it to move to the next binding site. And the next binding site could also be behind it, because you have a regular spacing in microtubules. So if there are at regular intervals, you can have Dynein binding sites. Now, if this is true, then it is also true that you do not really have a true step size for Dynein. Unlike kinesins had a regular stepping size of 8 nanometers. For Dynein, you cannot have this regular stepping size, because it can go 2 steps forward, then it can have 1 step. So it could be 8 nanometers, it could be double of that nanometers, it could be 16 nanometers too. So because of the asymmetric walk, the Dynein stepping size is not regular. You do not have a regular step size for Dyneins, unlike kinesins. So far, do you have any questions? What I will do is, in the next class, I'm going to repeat the same ideas that I have presented today, so that these ideas get reinforced, and maybe I can take an example from the papers that have discussed this, so that you are able to better visualize it. It's all a matter about visualizing how dyneins work, because they're pretty complex. You have this entire single polypeptide where one activity of ATPase hydrolysis is at one end moving these two coil domains that are attached to microtubule binding ends. At the same time, because of the linker attached to the tail, you have a movement of the tail. The tail is also pulling along the cargo. So you have, at one point, you are stepping on the microtubule, the dyneins, and creating new sites for it to move. At the same time, it is pulling along the cargo and pulling it along with it. So, in that sense, the movement of dyneins is actually pretty complex, but if you think about myosin's, then it kind of works very similar to myosin's. So, if it is bound to ATP, that's the point where it detaches from microtubules. And ADP plus PI state, that's the state where it has a weak affinity for microtubules. The moment the PI is released, that's when the affinity also increases. You have the creation of the power stroke, you have a change in position of the linker that can actually pull along the cargo that it is carrying. And unlike kinesins, and also unlike myosins, the stepping size of dyneins is quite variable because it doesn't have a regular walk. It's not a symmetric walk. It can move forward, it can move backward, it can move 2 or 3 steps forward before the other arm catches up. So step size of dynein is highly variable. Let me see if I've missed something for today. Okay, I think this might be, yeah. So this is a very beautiful video of a dynein that is walking on microtubule. I just put it for the sake of fun. What it shows here is that this track is actually a microtubule. In purple, you see the dyneins that are moving on the microtubule, right? And in green, what you see is a protein that marks the plus end of a microtubule, which shows that this dynein is moving towards the minus end of a microtubule, right? So with this, I think I'll stop for today. But if you have any questions, I'll take it. What I will do is, as I explained, in the next class, we will look a little more into how the dyneins walk, how they function, so that it becomes clearer to us. Maybe with the help of a few papers that have discussed this.
Hello, ma'am.
So, yes.
Ma'am, in the cell, how stability is generated in the cargo movement by Dynein. What I mean is it possible that another factor such as chemoattracting signaling can induce the cargo movement and thus to specify the Dynein movement? So it cannot go backwards, but it will only go the forward.
You're asking if there is something that would stimulate the cargo movement so that it only goes forward and does not go backward.
Yes, ma'am.
We will look into this.
Okay, ma'am.
We will look into this. Of course, there has to be something. Otherwise, how are you looking at this directed motion of dyneins, right? There must be some mechanism that is
Yes, ma'am.
preventing it from taking those few steps backward. But as I said, dyneins have the propensity to do this. They have the propensity to also take a few steps backwards. But that does not mean that, you see, dyneins is not something that we quite understand. This is fundamental to understanding dyneins, is that we do not understand everything about dynein. Okay, this stepping size that we see, this propensity for it to move backward, that does not mean that the other that the what does it mean? It means that suppose you have this cargo, you have two of its legs, and one of the leg has moved, say, one step backward, but the cargo is still here because it's attached to another leg, right? And this leg is also moving forward, so it has moved a little forward. But is there a mechanism by which you would be able to suppress the backward motion and promote the forward motion? We will look into this.
Yes, ma'am.
Hello, ma'am.
Yes.
Am I audible?
Yes, yes. Let me switch off my mic.
Ma'am,
Sometimes that
as you
gets it.
said that, ma'am, as you said that Dynein has six ATPs. So during ATP activity, all the six ATPs are active, like during the cycle of the movement.
Can you type what you're saying? I could understand that you have a question on the Dynein ATPase, but I could not get your question. Are all six ATPase involved in movement? Well, as I said, the ATPase function is linked to AAA1. That if you mute, so how do we understand, or how do you analyze, as researchers, how would you analyze which of these of the six subunits is important for ATP hydrolysis or important for movement? So the simple way is to you mutate. You start mutating the ATP binding residues on each of these, and you see what effect it has on the functioning of the dyne. So when they did such a study, it turned out that AAA1 is the main site for ATP binding and hydrolysis. If you mutate that, then you kill the dynein function. AAA3, if you mutate that, you slow down the dynein, but you do not kill the dynein function. So if you want to create a dominant negative kind of mutation, or say a dynein, a defective dynein, you would target AAA1, not AA4, 5, or other AA's, AAA subunits, subdomains, but the AAA1 subdomain. So this all of this idea comes from mutagenesis analysis, where they have mutated residues, specific residues, and then they have checked what is the effect on the movement of these dyneins. So you mutate a residue, then you see if it is moving. A simple, straightforward assay. So for the minors, you can refer to any standard textbook. What I have done throughout the lectures is that I have sometimes gone back and say when I say a particular thing, I have perhaps referred to some papers that have done that. But the general idea is there in any textbook. You take up any standard cell biology textbook, you will see the general ideas there. As I said, the exam will be open book. Yeah, they do not have the details because they will not give you exactly कि देखो इस एक्सपेरिमेंट में ये दिखाया गया है. मैंने शायद आपको वो एक्सपेरिमेंट्स बताए हैं कि इस एक्सपेरिमेंट में देखो इस तरह से करने की वजह से हम कह रहे हैं कि this binds to ATP या फिर this does not hydrolyze ATP. And this kind of detail is perhaps not there in the textbook, but it is there in my in the slides. You will see references for everything that I have said. Importantly, what is important for your minors is to remember this is an open book exam. There will be nothing that you can refer to the notes, you can refer to textbooks, you can refer to papers, you can refer to online materials, search it out. You're allowed to search. There's no issues with that. Your minor exam would be more like an application based. So do not get scared. If you have listened to all the classes, you will be able to do it. If minors may do not interact with each other, because if I see that any two of you or three of you have the same answer, then that is a red flag. That will not work. You can consult any textbook, any online material, anything, but do not copy from each other. All right. Okay, thank you, students. We will continue with Dynein because, as I said, this is, when you work with something for very long, you also have a very good understanding of it. And I have no shame in saying that I've not worked with Dynein, so my understanding of Dynein is also limited to what I read. And because my understanding is limited, perhaps my way of explaining it is also limited. So I'm going to try and redo this again maybe with some other क्या कहते हैं, some papers that have done this to reinforce the idea that we have obtained today. So whatever I said today, we are going to revisit it. So up to today's class is what you will have in your minors. Do not get scared of your minors. पूरा open textbook है, आप online search कर सकते हैं, सब कुछ कर सकते हैं. It will be very much kind of, I might ask you to design an experiment, or I might ask you to analyze one data that you have and tell me what it means. Something like this, very simple. And either 45 minutes या 1 घंटे का एग्जाम होगा. Open textbook, open materials, everything is allowed except copying from each other. You should not copy from each other. And I will send you a mail with all the instructions for the minors soon. The minors, the exam question paper will be available on Moodle. I will also send it to you by email. Okay. All right, students, if you have any questions, I'm here for the next 2-3 minutes. Stop sharing. Oh, no, that's okay. It's my job anyway. मैंने 2-3 classes तो लिए ही नहीं है, so we are kind of lagging behind. But I would not like to think of it in that way. Let's try to understand whatever we have learned so far, and then we will see. Okay, Friday there will be no exams because of gate duty. I will not be. I will actually not be available. So Friday there will be no classes. On Tuesday you have your exam. They will start at 2 p.m. So all the best. And if you have any questions regarding anything that you've studied so far, you can always drop me a mail. Thank you, students. I will upload the class slides on Backpack today. Bye bye.
Thank you.
Thank you, ma'am. Bye.